Let's start the lecture of nucleic acid conformation. Today, I talk about the local nucleic acid conformation binding protein. First, I will introduce about the bent DNA binding protein. What is a bent DNA? I show the structure of B-form DNA and the bent and the kink DNA. Usually, B-form DNA is a straight structure, but bent and kink DNA is quite different. And the most important, why the DNA structure shows such a bent and kink structure. Most important is that the Y B-form DNA is stable. B-form DNA consists of the hydrogen bonding between each base and the pi-pi stacking. And what pi-pi stacking is most important to form the stable structure. But in the case of bent and kink DNA, such a pi-pi stacking between the bases is disordered. Please see this smooth bending into the major group DNA structure. This is the X-ray structure, and this is a cartoon of this X-ray structure. This cartoon shows each pi-pi stacking of bases. In the case of B-form DNA, the distance between the bases is the same. But in the case of smooth bending DNA, this side distance is wider than this side is closer. It means that the pi-pi stacking between the bases is disordered. Please see the kinked DNA into the minor group. This pi-pi stacking is almost same to the B-form DNA, but this pi-pi stacking is quite disordered. It means that pi-pi stacking is very important to form the B-form or bent and kink DNA structure. Where is the bent and the kink DNA structure in the genome? This is the X-ray structure of nucleosome. You know the DNA nucleosome shows the double-strand DNA bind to the histone. It means that we can see some bent DNA in the nucleosome. It likes a smooth bending. This is a cartoon of the structure in the promoter. In the structure of promoter with the protein, tata binding protein bind to the promoter TATA site and induce the bent DNA. Tata binding protein is very important for the transcriptional starting. Tata binding protein is tata binding protein first bind to the promoter site and induce the kink and bent DNA, and then next transcriptional factor TAF2B can bind to this site because the tata binding protein induce a bent DNA, and TAF2B recognizes such a bending structure. And then finally, polymerase 2 bind to this basic transcriptional factor complex and can start the transcription from the promoter. This is the X-ray structure of tata binding protein with promoter DNA. And this green side shows the phenylalanine in tata binding protein. This phenylalanine insert into the TS base pairing and AG base pairing. And this phenylalanine can bind to the base by paper stacking. It means that the paper stacking between the DNA is broken, and then this DNA structure is bending. It means that such a paper stacking of the aromatic region is very important to induce a kink and bent DNA. This slide shows the structure of the enhancer and the promoter. You know that the function of promoter is that the function of promoter, function of enhancer is to promote the transcription from the promoter. But the major case, the distance between the enhancer and the promoter is very far. So, how to enhance the transcription from the enhancer? For example, such a DNA bending protein binds to the middle point of enhancer and promoter, and then the distance between the enhancer and promoter is very close. So, group of mediator protein bind to enhancer and promote the recruiting of the transcription factor binding to the promoter. And then this complex can start the transcription. It means that this DNA binding protein is very important for transcriptional starting. Now, I will explain about the gigotoplex binding protein. I already introduced about the gigotoplex. Gigotoplex is formed at the GGH sequence, DNA, and RNA, and this is the structure of C-mix gigotoplex structure. And especially, we can see the lot of gigotoplex structure in the promoter and the telomere region. And not only DNA but RNA can form the gigotoplex in this region. And this is a gigotoplex binding protein. And we know that a lot of kind of gigotoplex, but almost the gigotoplex bind to the telomere region and the promoter region. Especially in the telomere region, POT1 is very important for the telomere maintenance. POT1 can bind to the gigotoplex in telomere and control the telomere length. And in the case of promoter region, this nucleolin is a very important gigotoplex binding and controls the transcriptional level. At first, I explained about the gigotoplex in the telomere region. In the normal cells, telomere length is shortened, and then normal cells die. But in the case of cancer cells, telomerase is active. It means that this telomerase can elongate the telomere length, and then cell becomes cancer cells. But gigotoplex inhibits the activity of the telomerase. Telomerase usually bind to the single-strand DNA and can elongate the length of telomere. But if telomere region forms the gigotoplex, inhibits the binding of telomerase. In the cells, port 1 bind to the digitoplex and unfold the single digitoplex and then produce the single-strand DNA. It means that the port 1 accelerates the telomere elongation by unfolding the digitoplex. Not only port 1, but the TLS1 and the TR2 is a very important telomere binding protein, and this protein can bind to the digitoplex, but not unfold. This protein folds the digitoplex. This digitoplex binding protein binds to the digitoplex DNA and the digitoplex RNA in telomere region, and then induce heterochromatin in this region. This heterochromatin region in telomere is very important because this heterochromatin region inhibits the recombination in this telomere region and then telomere shortening. It means that these digitoplex binding inhibit to become the cancer cells. You know that not only telomere region but the promoter region can form the digitoplex. This is the example of the thymic lesion. Thymic lesion is one kind of cancer oncogene. Transcription factor bind to the promoter in this thymic lesion. This thymic is transcribed, but this lesion consists of the Z-lead sequence and can form the digitoplex, and then nucleolin bind to the digitoplex and inhibit the transcription from this lesion. It means that this protein nucleolin digitoplex binding bind to the promoter in thymic lesion and inhibit the transcription from this oncogene. Next, I explain about the eye motif. Eye motif consists of the C-lead sequence, and almost eye motif forming sequence is in the promoters. Thymic and BCL2 is one kind of oncogene. It means that this eye motif can control the transcription, and this structure has an important function in the transcriptional controlling. This slide shows the relationship between the digitoplex and eye motif. And you know that this lead sequence can form the digitoplex. And you know that double-strand DNA has a complementary sequence. And this C-lead sequence can form the eye motif. It means that D-lead and C-lead promoter can form the not only digitoplex but the eye motif structure. HNRNPLL is one kind of eye motif binding protein. Especially this protein has four kinds of RRM motif. RRM means RNA recognition motif. This RNA motif can bind to the eye motif and unfold this structure. This HNRP can bind to eye motif in this specific promoter region, and this promoter can promote the transcription from this promoter site. It means that HNRPLL promotes the transcription at the eye motif containing promoter.